Back to it's something that will require attention and some kind of. Did we do? Yeah. So we are going ahead now. Thank you so much. Wonderful. It was short and sweet. Very nice. So yeah, this is your mic. I'll. You can hear me like this. This is Vivian. So Surjit's. I told his story. Huh? He got. Yeah. 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 So Vivian was my master's student in VIT. We know each other from there. About 2008. 2008 onwards. So yeah, his class students, uh, uh, they had uh, very. They tried to do some campaigning and all against me, right? That all. Yeah, yeah, as usual. Yeah. So he did two courses with me, right? One was on enzymology and another was on proteomics. Yes. Yeah. Vivian was an original guy. He never rote learned. He never got great grades, but he 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 knows how to search, and he thinks very clear, very simple, very free. And I have great respect for him. We are He's my spiritual guru and all that. If I have some problem, I talk to him. Basically, yeah, you resolve things for me. It doesn't mean that just because you're age-wise junior, it doesn't mean that all that. So, but then if he says something which doesn't appeal to my nature, I can always say, I'm the boss, I'm the guru. So that is always there. Yeah, he has a very good understanding of music, and he plays violin very well. Yes, huh? Yeah, he has good understanding of music, and he plays violin very well. And he generally has good aesthetics. I've got very good opinion about him. Vivian, all is yours.
Thank you. Thank you, sir. Thank you for this opportunity as well. So I'll just take a couple of moments to, give you a brief idea about our interactions. Like you said, it started in 2008. But more specifically, I would say we started interacting more towards 2017, 16, that time. So what you've been seeing here today is probably, a culmination of a lot of years of hard work. So you might, people who come across, Professor Murli sir, as I call him, have sometimes very polarizing opinions about him. And, for many, he might come across as an overconfident person, to some as an arrogant person, some as too much of a pessimist, for some too much of an optimist. So he generates very polarizing opinions about himself. That I think he would agree as well. And what you see here, right, all these equations, all this work that has been presented, most of it which I myself not able to comprehend, a lot of the work as well. I think at least some of you can relate. But what I want to put forward is all this stuff did not happen overnight. And a lot of it, I would say, happened over, many sleepless nights, which I've seen him go through. So what is presented here is just, we are seeing the top of the iceberg. So I've seen him work throughout nights and nights and nights together without sleep, and maybe hardly sleeping, hardly 1 or 2 hours some days. And, we must have an understanding that whatever you're seeing here is a result of all that work for a lot of years. And if I may say, at that point of time when he, really started working on it, we might all have excuses in our life. I could see him, without funding, without any motivation. If at all there was any motivation, it was all the rejections which he received from, all the publishers. So I've seen him go through that grind for, I must say the initial years were very, very tough. You can see all these papers published, but what you have not seen is the rejections that he got, the mean letters, the shunning of his work, criticisms, and that too false criticisms which he had to deal with. So all of those efforts actually culminates into these publications. And what I've learned seeing him these past years is, if I may give some sort of learning that I've acquired seeing him go through this process of doing science, is keep the child in you alive. Ask simple questions. Keep the curiosity alive in you. So I have a young son myself.
That is also like that.
Okay, it's great to see similar minds. I have a young son, and if you look at children, things that we take for granted, they might ask why is it like that, or you can see them wonder why is it like that. So if you look at some of these protein structures and all these things and chemistry happening that way, if we study from a textbook, or if we learn for our exams, okay, we might look at some equation or the C scheme or phosphorylation. Okay, ADP gets phosphorylated, we get ADP. It's as simple as that. But maybe sometimes it is good to stop there and ask, why should this happen? Or why should this happen like this? When we study, we just study it. But I would say, think like a child. So if you had seen those protein structures which you saw, those complex 1, 2, 3, 4, 5, why are they like that? Why is the shape like that? Why is the protrusion like this? Why? So that is one thing which I've learned. So be curious, keep the child in you alive, ask simple questions which appeal to you from that. So when I say this, I still remember, I think 2016 or 17, the day he called me with, I was wondering, what happened to this person. He's a professor, and I know him as a professor. He's calling me and screaming like a child that, I found this, Vivian, I found this, I've solved this. And, obviously I had to act excited at that moment, but I was just wondering what is happening at the other side. But and I think maybe even some of you would have felt at least some goosebumps when he showed you the calculation of how many protons there could be in a mitochondria. Something, it's just +2 chemistry, I may say so. But we haven't, thought, how can this happen? So he keeps that excitement alive in him, that childlike curiosity. And for those of you who are pursuing science right now and who want to take it up as a career, one advice, if I may say, or give you, watching his progress through this, is keep the child alive in you. And when I say all these complex theories and equations which we saw here for these couple of slides, he didn't right away one day sit up there and think, okay, I'm going to calculate the delta G for this equation today. Or he didn't question that this chemistry or this electron transfer doesn't seem right. It did not start like that. So how it starts is with very childlike curiosity, and how it starts with is very, very, very simple questions. So coming to the vision part of it, I'm not a chemistry expert or a person pursuing science right now. I work for a private company organization, but let me give you an idea of of my learnings from him through a couple of slides. So this, as you can see, is the structure of an eye, a very rudimentary one at that. And have you, thought about what happens when light enters our eye? Or you would have a basic idea as students of science that light enters through, our pupil, and it forms an image at the back of our eye on the retina. So just a couple of questions, if I could
Sure.
put you to think through. So when light enters through our eyes, does it straight away go to the retina, or do you think there are other objects in the pathway of light? Sorry? Okay, there's a lens which focuses the light. So does it straight away go and form an image on the retina? Sorry? Nerves? Yes. Cornea? Okay. Okay. What can you see from these images?
What he's asking is, this is the structure. There are the rods and cones which are supposed to photoreceptors, right?
Yeah.
So does the image directly go and fall on the photoreceptors? That's the question.
It doesn't. So what does it pass through? It has to pass through all these cells in front of it, right? And then there is the liquid portion of our eye. What is it called? Vitreous humor. So all these things are there. Let us, just suppose that vitreous humor is just like glass, it passes through. But even if that is the case, all these cells are there in front of it. And do you think that passing through all these cells at that density, the light which enters our eye, or the image which enters our eye, will form an image like what you see here on the screen being projected? Would it be like that if I come and stand in front of this? There is obviously, I'm an opaque body, but there is obviously some disturbance which you see on the screen, right? And even if it does fall on the retina like that, how do you perceive depth? If it falls on a surface like a retina, does it form a 3D image or a 2D image? 2D image? Are we sure?
It forms a 2D image.
Okay.
The brain processes the difference between the two images to form a 3D perception.
How does it do that? So that will come to the chemistry of it as well. I wouldn't be covering the chemistry, but so these are all questions. So what I'm trying to say here is all these theories which he is saying, the murburn concept did not just get formed in one day, or it didn't form as a result of complex thinking or calculations. It is just simple, very, very simple questions and childlike curiosity which leads to it. Just simply asking why. So as you can see here, there are many layers which it passes through. And then one more thing which, if I may say, is numbers. There's a beautiful book which he introduced to me called Biology by the Numbers. Excellent book if you want to have an understanding of, sizes, shapes, etc. of, our cells and, biological stuff. So if you haven't come across this book, please do. It's called Biology by the Numbers. So just another question there at the bottom, if you can see it. So a 400 nanometer photon must travel, if you see this distance of 400 microns to reach a cone cell. That's in between that, there is diffraction, reflection, diffraction, scattering, all these things are there. So how is the image formed? Is it just falling directly on the retina, and then, as they say, is that leading to a cis-trans isomerism, which is making the signal? Or are there other possibilities for this? And when we start asking why for each and everything that is being taught to us, we will start searching. And to search or to start searching, there is an admission which we must make. So you might see him confidently speaking, here, but that confidence comes from questioning himself of what he knows. That admission of that I do not know. So we can only proceed from there. So stuff like this, very simple questions, which leads to theories like the murburn concept, and which revamps our understanding or deepens our understanding of how life processes work. So, for example, when we black out, right? When we lose oxygen, why is that? So we start thinking about why is oxygen playing a role in how we see? What is oxygen's role there? Then the speed at which we process images, right? How is our neural system capable of processing images at that speed, with considering the wavelength and considering the neuronal conduction speed, etc. Then how is, like you said, right, our brain just processes the 2D image into 3D image, things like that. But how does it do that? How do we get the depth perception? How is that signal actually formed chemically? Then, there's a resolution factor of it as well, which again brings us back to our, question. So coming back to the chemistry part of it, I would like to hand it over to Murli sir. But, just to give you an idea of, how theories like this are formed and what could motivate you as students of science to pursue this as a career and be really successful. Be like a child, be curious, ask simple questions. Never hesitate to ask simple questions. So, thank you.
Thank you, Vivian. Thank you so much. Yes, switch it off, please. Yes, can you hear me now? So, let me just go back. So, basically, here, we know enough data has accumulated. Pardon? Can you hear me? Yes, it's there. So, now, here, in the physiological experiments, they have seen that when you put more light, you get more ROS. Now, did we come across some such observation in respiration thingy? Do you remember the Nichols paper, the image that they reproduced from aging cell? It showed that more ROS, you give more NADH, you get more ROS. You give more oxygen, you get more ROS. So in this physiology, more light intensity, you get more ROS. So that's a very important question. Why should visual process have that? Then why should, when we know that the RPE cycle, retinal pigment epithelium, that is so slow and wasteful, that is the cis trans isomerization pops out the retinal, and then it has to go through the retinal pigment epithelium. And one second. Oh, okay. Let me see the order. So the questions were asked with an assumption that you know that. So I will come to the classical model. So then you know what is a myelin sheath? Some neurons have cover, and that you have faster connection. So if I would not imagine if that were the case for the classical neuronal conduction. Then conversion to photochemical cue. How is the transmembrane potential variation affected by isomerization and ejection of retinol and all that? So why does rhodopsin get phosphorylated or oligomerized? Is that oligomerization something that rings a bell?
Limited
No, oligomerization. We came out in hemoglobin, in LDH, in so many respirasomes, in the photosynthetic proteins. They kind of, why is it that happening? Then, yeah. So sometimes what happens is this is the curvature. The arc value that you have got is that it's based on the angle subtended and all that. The calculation basically, one rod cell can basically cover this much area. So based upon the distance, you can only the amount of points that it can resolve. Our eye has got much higher resolution than the dimension of a rod cell. Did you understand that? That how is that resolution possible if one layer of rod cell is the fundamental phototransduction unit? So questions like that. So let's see what is the classical. Now before we go there, we just studied what one? Photosynthesis, right? Photosynthesis, what happens? The photosynthetic procedure converts light energy into some chemical compounds. Now, what does light energy do here? Yes, it transforms that light energy into some kind of tangible signal that our brain can process. Yes or no? All of us agree that. So now the point is, look at the classical. This is the visual cycle for which the Nobel was given early, right? So rhodopsin is the protein located in the retina, that is specifically in the last layer of the eye, this layer right here. So made of rod cells and cone cells. So there is this guy. Where did you see it? Did you see it in photosynthesis also? What are the pigments in photosynthesis? Carotenoids. I told you during that time there is carotenoid, that is, and I told you Schiff's base. Did you remember that? Yeah, there was some, and I told you that pH was 13, so it cannot pump protons and things like that. That was a bacterial rhodopsin, but this is our rod's rhodopsin. Now here, then what happens is, this is a light active organic molecule which is embedded in a protein, right? And that protein, when light hits it, this from cis it goes to trans, all right, and there is a bunch of chemical reactions that sets in. So now we are coming to how a cell responds, right? And earlier part was how the metabolism was. Now tomorrow we will go to how the cell moves. Okay, mechanical activity. Okay, so that mechanical activity, here is an electrical activity, a response. So that is yet another type of response. So the point is, based on non-intelligent, not highly selective processes, how can a cell make basic responses so that it will serve its cause as an organism? Now, here is the basic component of the cellular visual cascade. You have rhodopsin here. These are all the rod cell at the distal part. This is the retinal pigment epithelial cell. These are the rod cell. Now, these are like the chloroplast, exactly like the chloroplast, exactly like the mitochondrion. You have convoluted membrane structure folding over itself in the rod cell. Did you understand this part, the similarity? Photosynthesis also has carotenoids. This also has. Now, here, the basic reaction is that when one light photon hits, the cis-trans change happens, and then there is a chemical formation of a bond and all that, and then that pops out the retinal. And this sets a sequence of cascading reaction. What's a cascade? Have you seen dominoes cascades? Yeah, you know that? So how a small one photon can give us a electrical activity that in turn can be conveyed. That's what we are focusing on in this talk. All right. So this this transducin, now you have a membrane. You have a membrane like this, a photon coming and hitting. This is the rhodopsin. The rhodopsin is attached to a membrane transducer, if one may call it, called transducin. Light to electrical energy impulse transduction. And then there is an exchange of that protein is bound to GDP. It exchanges for GTP, leads to activation conformation change. Then phosphodiesterase 6 transducin detaches from here, goes and binds with phosphodiesterase, initiating another set of cascade. Now, do you see something similar that we have already questioned earlier in many? The deterministic nature, and then what else? What else, Mohammed? What else, people? What else? You know, one protein is going to another protein and kind of binding and all that. All that. Right? So then it's a wasteful thing, and then this takes hours. Now from transretinol, it has to go to retinol, this enzymatic sequence that happens here, and then it has to come back to the outer segment. This takes hours. Now, but then this rod cell is easily available for within millisecond time scales. It is re-available. How? Time. It takes hours and it is ready in seconds. You know, that elephants coming from Kerala to, not Maharashtra, sorry. Mansarovar, yes. Mansarovar, that's a lovely place. No, not that Maharashtra is not. Mansarovar. Yeah. So, now, say, sir, here, this is bound to a protein, sir. Now, when light hits that protein, what happens is a set of, and this pops out, this thing pops out, it has to go back and come back. So, these are all one photon. This is hours, this is milliseconds kind of time scale. Sir, all this is crazy. So, please, you have not seen the kind of crazy things we have already discussed. So, basically, this does not make basic quantitative sense if one were to. So, and then the signal relay is via an ion flux. What is an ion flux mechanism? Have you heard of Hodgkin-Huxley's mechanism? That is, sodium goes out, potassium comes in, channels close, channel open, everything that goes, and then it is going like a ripple along the nerve. So that is, and then this rod cell fires. What happens is, there are two states, at rest, at activated. Rest, the calcium ion channel is closed, cyclic GMP. Now, otherwise, when it is activated with light, transducin goes and binds to phosphodiesterase. That is what is shown here. Okay. Phosphodiesterase, GDP transducin, it becomes GTP transducin and GMP. All right. So this is the basic reaction. So like P450 system, like photosynthesis system, this is multi-molecular deterministic scheme. Yes. So that is what is shown here. Rhodopsin, light, some chemical changes occur, and then you have a skip space formation, and then all transretinal, and then GPCR activation, then GTP is exchanged for GDP. Why should GTP be exchanged for GDP? Why this? Why? Because some people said it exchanges. Why? We don't know. All right. So it exchanges GTP, then GTP, this transducin, active transducin, it goes and phosphodiesterase binds, and then the phosphodiesterase which was inactive becomes active, and then that's the way of the cycle. This is the visual cycle. Questioning the classical perception. Now here are the structures of the protein. All right, here are the structures. This is rhodopsin, and the udukupola, like it's a protein like this. It's a protein like this, and just rhodopsin just sits just somewhat in the membrane side, and this rhodopsin per se across the living beings, it's not structurally conserved. There's a significant variation in the structure of rhodopsin. So if you imagine all these conformation change based, all these deterministic things, you would imagine like I showed in cytochrome C or in plastocyanin, there was no conservation. All this, if it is so specific, you will imagine. And then this alpha is supposed to detach and go and interact with phosphodiesterase, and then there are supposed to be some sites that are not free, and that is supposed to be available and all that. So there is data known that alpha transducin, when bound to GDP and GTP, this is an image generated by Danny, the structures are almost identical. There is no difference. So why is there a change for GDP or GTP? What is the activation? There is no scope to explain. So and what is the role of this guy? There's a huge thing sitting here. All right. So this is the structure. This is the whole thing. This is the basic. There are some holes and all that here. We don't know what their functions are. There is no role for all that. Like in hemoglobin, those apertures and all that, no role, right? Why GTP, GDP exchange? Nothing, no role. So murburn model of vision. And most importantly, there is no direct role for oxygen in vision. In vision, most importantly. In the murburn model of vision, the retinal can get activated and give electron to oxygen. All right, that is a fundamental process. And then the same Ross dynamics can regenerate. But what happens is this effective electron transfer, I'm not going to the electronic states and the various complex chemistry aspects, but the fundamental electrical activity is what? Assisted by oxygen, what retinal does upon receiving light is it gives electron to. This happens anywhere. You put retinal here, you put light, you give oxygen, it will become superoxide. The protein does not have any magical powers. It need not get conserved, cisretinal, transretinal. It was just an observational coincidence. In P450 cam, it bound, it was changing, something like that. So, something like that was the reason why that cis-transretinal became the active site mechanism. So, E-transfer from retinal leading to the formation of ROS in the vicinity of rhodopsin. The lost electron can be recovered DOS-assisted reaction similarly as summarized in the right. So that's what is summarized. So here is the murburn catalytic cycle, that is active rhodopsin, photon pinches, oxygen becomes superoxide. Now this gives electrical activity, yes or no? That is, then this attacks phosphate and GDP to make GTP, right? Like in the ADP synthesizing process. Now GTP the transducin, what it does is it hydrolyzes GTP, releases GDP, which is a small molecule. GDP goes and binds to phosphodiesterase, and then you have an active phosphodiesterase which can give the subsequent cascade cycles. I'm not going into the cascade part. I'm just sticking onto the basic phototransduction procedure. Now what? How does it look? Rhodopsin, light, basically activated, plus superoxide, plus positive radical form here. Now there is no wasteful procedure here. It doesn't pop out. It doesn't have to go to the retinal and pigment epithelium. What they saw was the cis-trans was many times when it goes, some can get deactivated and pop out. Like what that day he was saying, after multiple cycles, the protein can lose certain things because certain activities might lead to that. So this is a radical involving reaction. So transducing inactive alpha, the ROS can attack that, making GTP. So that GTP is not having affinity for alpha transducin. It comes out and binds to the beta transducin directly, and then that beta transducin is a GTP hydrolase, and that gives GDP. GDP goes and binds to the phosphodiesterase activating, leading to the subsequent cycle. Here there is no multimolecular deterministic scheme. Did you understand that part? Simple bimolecular reactions and electronic cascades. So basically here is the here it is how you don't need protein detachments and going selective bindings. You see here excitation gives ROS formation and retinal excitation R plus, then GDP is basically made, GTP is released. This guy takes the GTP, hydrolyzes it, gives the GDP, and like that. So it's a very simple, quickly it can recycle. Then and there is no wastage. All right. Now, how is the relay? 1 second. Where did it go? Yeah. How do we support that? The slides got a little scrambled. Yes. Pardon? This equation, you are saying something? Where, sir? Which one? So basically, see, we are hydrolyzing GTP. So it requires a hydrolysis, requires one water molecule. So that GMP, where GMP, what this is also cyclic GMP. It has to become. It means it is water requiring reaction. So this is the overall. In this cycle, oxygen is needed. No dissociation of retinol. GDP signal for phosphodiesterase 6. Alpha transducin is a GDP phosphorylase, and beta transducin is a GTP hydrolase. So now what is the evidence for that? Here we predicted the KD of GDP is for alpha transducin and beta transducin. Here it is. The simple docking that we did clearly shows KD of GTP is 0.5, 0.6 millimolar, whereas KD of GDP is picomolar ranges, 80 picomolar, right? Now, so what? GDP will be strongly bound at alpha transducin. So when you see the structure, when this thing comes, ROS comes in and goes through the aperture into the alpha, attacking GDP, and here you will form GTP. As soon as GTP is formed, will it have affinity for it? No, GTP comes out. So when this comes out, the structure beta takes it. Beautiful. So here, the beta is shown here. So GDP high affinity, GTP low affinity, and then here for GTP high affinity for beta. Wonderful. This was predicted by me, and you did the docking, and we get it. Very simple. And the molecular structure is beautifully explained. See how it is bound, all that. Yes. Pardon? Yeah, excited. No, the electron has gone away from here, no? So it is plus. Yes. Yeah. So, but this has basically some light can give, it can give light gives the energy, it can excite a molecule, it can give an electron, then the electron, after that it will, superoxide can react among themselves, become peroxide using water, proton, and then you will have the recycling like that. So basically here you see we have a very simple role for beta transducing that explains why GDP is bound to alpha, why GTP is used by beta. The earlier one, they didn't know what beta transducing does, the binding. So what does the experimentally observed rhodopsin oligomerization do? It enhances radical-based functionalism. All right. So it explains non-conservation, non-essentiality of the protein fold across diverse species. The rhodopsin transducin, sorry, this G was not required, beautifully agrees with the murburn model. So here I have no evidence, but this is a very simple model that explains how the when a neuron takes a signal, whether it is a chemical signal or a pressure, what it does is it messes with the oxygen superoxide equilibrium, and that is a chemical signal. So electrical signal. So basically that's what it does. And so here the structure is there is a definite polarity in the structure, and that this can lead to a electron relay in the kind of order that we want, we expect, and this polarization, hyperpolarization, and all that events, this electron transfer. The current theory is ion pumping, sodium potassium. I am saying electron transfer. What they did is Hodgkin-Huxley observed, put a electrode here and electrode inside, and then they observed some peaking phenomena like this. So they said that sodium channels are there, potassium channels are there, there are pumps, there are channels, some pump, some open, some close, something like that. And they explain the potential differences. Now based on pumping. Now one of my collaborators can also get exactly the same nature of the equation, explaining this kind of mathematical behavior, but not based on pumping or selectivity process of the membrane, but due to adsorption process. So the constants are different, but you get exactly same mathematical form. You remember I put a hyperbola and I showed you multiple equations can give the same experimental fit. So that's what you are seeing here. So I'm sorry, Hirohisa was supposed to be delivering this part of the lecture, but his son suddenly took ill. So that's the reason. So I want to see if all the slides are covered before. Yeah. So now from here, what really happens is from here to here, there's a polarity. Here what happens is oxygen superoxide equilibrium. Now this oxygen superoxide equilibrium, whether pressure, if I touch, this can get affected. In chemical, so many things, this can be a simple mechanism for the electrical activity. And the oxygen water superoxide, all this equilibrium can be a way of recycling, resetting the neuron also. So here the retinine pigment epithelium rod and the overall connectivity of the intermediate cells can be explained by a simple kind of semiconduction or some kind of a model like that, wherein here, rather than the rods being the primary phototransducers, the earlier cells in the overall eye structure, here they believe that this is I say that all these are phototransducers, and this is actually supplying the electron deficiency by effective charge separation. So you see, it's what do you call, nowadays there's this modern day LED things, no? Am I right? From the back there is a light projection, and with respect to that there is a visualization. So this is what happens here. So now, yeah, so brain is receiving electrical signal. So this is, rather than an ionic flux propagation, this is an electron transfer, because this neuron has got defined polarity. Now,
Sir, I have one question.
yes.
How do you propose the electron moves across?
Yes, that's a very good question. I have no very defined idea. I think this membrane can be one way, or it can be through the microtubules also. I told you I have no data here. This is a very simple model. See, the other model is the Hodgkin-Huxley model, which says that sodium potassium, how can it explain that when it is covered by myelin sheath, the conduction is faster? You want the ions to come in, go out, go like that, right? Here you just want it to go like this. The radial dissipation, the lateral dissipation, nothing happens here. Here, this is a better explanation. Then the electron movement is explaining this circuit. There is a model for justifying the theory, but then multiple experimental data, so many of my collaborators and the history has always overlooked all the data and all the workers' objections against this model. Because the Nobel was given to Hodgkin-Huxley in 1950s, I have Federico Faraci. He's a collaborator. You just Google his name, Federico Faraci. You will see his master's thesis. He has just done masters. Wonderful piece of work. How a scientist should. He's actually from an engineering mathematics background. He has systematically questioned, and the data produced by not just one, multitudes of Japanese, all those scientists in Europe and America have systematically questioned this. But that is never brought to the light. I'm not going into the details of that because I want to also cover the electrophysiology part. So basically, what I'm saying is, until now, vision did not give any role for oxygen. There was no real tangible logic for electrical stimulus formation. We have a tangible. There was no logic for GTP, GDP signaling. What was the signal? Protein was supposed to go and bind. That is not. GDP was supposed to bind. And it gives a very tangible thing. After the thing happens, after a signal is relayed, it detaches, the reattachment thing, everything, resetting is wonderful, beautiful. So now here is the other theory that electrophysiology. What is electrophysiology? Electrical activity and also the electrolytes in a cell. How are they maintained? I told you that the cell has a preponderance of certain negative charges inside, that is explained as a pump in the membrane theory. There was another theory called association induction hypothesis, which is by virtue of binding of some cations onto this, you have a negative charge. That also can be a possibility. Then murburn concept, which says that there is effective charge separation, which leads to electronic charges here. So these three theories are very different theories, but this Murburn does not rule out this, but definitely both these theories question this. So my collaborator Lauren Jake and Hirohisa Tamagawa and the earlier other person like Vladimir Matveev, Gilbert Ling, all those people that were opposing this, Cambridge School's membrane pump theory, they differ in this. I'm not going to go into details of the number of agenda here that is clearly beyond the current interaction. Now, here is what the reason for the sodium-potassium difference is as per the classical theory. This is a molecule which is composed of two parts and three parts actually. This is the alpha part, which is like this. This is the beta part. They say that this protein takes in 3 sodium, then pumps the 3 sodium out, hydrolyzing ATP, and then it changes some structure and it gets higher affinity for potassium. When it is bound to phosphorylated form, it is formed, then it comes in. 2 potassiums come in. All right. So Freeman Cope committed suicide. This guy won, and because Jensku was given a Nobel for discovering this protein, which was supposed to pump it like that. They were already looking for protein pumps, and they were already looking for channels for which 2003, what is his name? McKinnon, right? Roderick McKinnon. He got the Nobel. Now the point is that if this did work as a pump, what do you expect? You would expect definite binding sites, definite logic for conducting them, definite structures, why, how, all that. So anyway, the first, now you understand the intricate mechanism it should have. This single module should have very simply represented. It should have ATP binding site, then it should take 3 sodium in, and then you see closing and opening. There are gates inside, out. The one gate opens, the other gate is closed, then this gate closes, then this gate opens, chucked out, then it should change affinity, because now the outside, it is a lot of sodium outside anyway. So next, the potassium is very low amount there, so that potassium should be next coming in. This same sodium should not come in. So for that, it should have a different affinity, and then it should take in potassium. So it requires the simple protein should have wide number of gates and very sequential arrangement, beautiful way in which it should all open, close, otherwise, and it should also be selecting all that. Did you understand? It's a serial sequential process, approximately 15 odd steps to pump out 3 sodium and to pump in 2 potassium. That's what is the reason. As per your body has high amount of potassium cells, that is, and sodium is high in the outside. Now that is the reason for this. Now your heart patients, sodium high, potassium low, all this profound impact on your working. Now they say this is because of pumping action, whereas Murburn concept says no, it is because of oxygen superoxide dynamics. Why? Where is that? Yes, you see here, very simple. The thermodynamics explain the states of increased potassium inside cell, See, you see, potassium and sodium, though they belong to the same group, their relative dimensions are given here in the angstroms, picometers. Here, you see here, sodium plus electron gives solid minus 2.1. This is fissile, whereas this is non-fissile. This is the distinction. It is not the size, it is not the affinity that is the distinction. Do you understand? So these are the different energy yields. I'll come back to that. So basically, and also you see carbonate, acetate of various ions. You see the solubility of chloride. If you see chloride, all are around the same range. Potassium chloride, sodium chloride. So based on sodium chloride, you cannot differentiate. But you see phosphate, you see acetate or carbonate. The cell interior, what are the respiratory anions? Basically, respiration leads to CO2's oxidation, oxalates, basic carboxylates, so many in the respiratory anions, and then phosphates, right? You have basic biochemistry, you know that, right? Respiratory. So when oxygen assisted reactions occur inside, you have respiratory anions, and proteins are they have carboxylate side chains, glutamate, aspartate and all that. They are generally negatively charged. So they will preferably take in potassium. You see this, say phosphate, solubility of potassium phosphate is 4.2 into 10 to the power 6, whereas potassium potassium phosphate, you see sodium phosphate 6.9 into 10 to the power 5. There is a difference in their co-solvation ability. So when the interior it's a negatively charged ion, phosphate, then what happens is, and the interior has high amount of phosphates, bicarbonates, proteins and all that, it will naturally prefer potassium. And the sodium in the membrane, this sieving of preference of potassium to migrate inside, here this is a lipid membrane, right? That is ensured by oxygen comes in, the membrane will, here in the vicinity there will be superoxide. This superoxide equilibrates better in here with potassium, and that's why it comes in, and the sodium does better with chloride. So that's the reason. So co-solvation and the redox dynamics is what is doing. So the key equilibriums are this, and here is the other aspect. So it is not a 3:2 pumping. There is no evidence for that. It has just come because of consensus. It is because of co-solvation. Murburn model accepts the cellular milieu to be a conservate and colloidal, considers TMP, that is transmembrane potential, to be a consequence of respiration. So how do we show that? We just studied the structure. The beta is not supposed to have, we don't know what this does. The people don't know. They say it binds, the potassium, sodium binds and transports here. Now, when we check the structure of the cap here, lots of arginines are hanging like that, right? Arginines are these arginine only, right? Arginines. So now when they hang like that, it enables a significant amount of superoxide stabilization there, right? So that's what it does. And see, you see the porosity of this kind of a molecule. There is no real channel, there is no opening, there is no closing. It is just their imagination, wild fancy, because that is dictated by their theory. You see the sodium and potassium and all the magnesium, all the ions that they saw in crystal structures, they bind at the same place. They bind at the exact same place. So how can they differentiate? There is no logic for differentiation. So here is the logic by which we explain the structure, that is preponderance of lysine and arginine on beta subunits enables it to serve as a ROS recruiter, porostructure of protein with multiple surface systems. This is a redox active protein. So how do we see that? So one of the most important cardiotonic medicines are plant and toad. You know this bufalin, that toxin that these Amazon people use on their arrow. Yeah, that it is supposed to go and bind here and very clearly block the channels and something like that. Now, if that were true, the steroid backbone is this. This is the basic steroid backbone, and here they have cardiotonic steroids. So this was supposed to go and block this part, this entrance part, beautifully. You understand this? This entrance is blocked, topi vachamari, this one. So you see, if that were true, so many molecules would do. Now what we are saying is that this bufadienolide and cardinolide is redox active. That does the superoxide mediate, that disrupts the superoxide mediated dynamics at the interface. So that alterate, you see how lithium is given to stabilize nerve activity in some patients, because while rubidium is the same as potassium, you see lithium has much higher value, so it will change the roles of potassium here. It will change that. It will outcompete. So simple NK structure and oxygen sensitivity, uncoupler. So basically, this protein has nothing, no redox active component, yet it is sensitive with lot of antioxidants like this. And if the steroid backbone was that important, how could we get these molecules, oxanol, citrioviridin? They don't have a steroid backbone. They also give the same activity. So that is because that binding based effect is not the outcome. The outcome is primarily determined by this kind of interfacial things. So then there is a transdermal peptide which exactly like the CPR. Remember, N term was supposed to come together. I said kiss. Remember, they were supposed to do that. Now that has also got lysine and arginine like that lot, and so that's messing this is a very selective inhibitor for that. It's a peptide inhibitor. So so what Hirohisa showed in his experimental lab based upon my inputs is that he took transmembrane potential measurements. He showed that an enzyme producing ROS can also give potential variation. Did you understand? Basic simple thing. So TMP is a complex phenomena wherein ROS can contribute, and the ions and TMP are not only the issues. So there are different systems. The different distribution are not giving rise to directly correlatable transmembrane potential values. These things are also dependent upon other factors. So there were systematic study wherein the ion differences were not really actually correlating with the transmembrane potential. Even a synthetic microsphere can give exactly the same kind of nerve conduction impulse, transmembrane potential fluctuation like the without pumps and ions. So this is simply a redox process and not an ion pumping process. So this is a very superfluous, and also plants don't have NKA. So I come to the last part. So these are the, see here, this is something very simple, single ion channels. The electrode is 1 micron like this. So one bacteria is 1 micron. So how much is an ion channel? 1 nanometer roughly. So they say it can give method to study single ion channels. 1 micron, single ion channel, give me a break. The electrode is 1 micron diameter. Ion channel is 1 nanometer. 1,000th of that. Do you buy this? I don't buy it. I don't buy it. So all this. So we have shown that all this is catering to the mandate established by the Cambridge School. So that's why I say after Second World War, Anglo-American interests got the better. And the scientific world, not in physics, not in math, but in biology, it is driven primarily by consensus. That's it.
Biosensor, they are using microelectrode, thickness of electrode is 1 micron, very.
Yeah, I am not saying 1 micron is not possible. What I am saying is, the Nobel citation is, it can allow the study of a single ion channel. What is a single ion channel? It is 1 nanometer. So now you have something like this. You are putting an electrode here, right? This is supposed to confine this area is 1 micron diameter. How much is the diameter of an ion channel? 1 nanometer approximately. So it will, and one bacterium is 1 micron diameter. Now this bacterium has thousands of pores. How can that be? Do you understand what I'm saying? No, very good. If you don't understand, you say you don't understand. So this is a microelectrode, right? Okay. Now let us magnify it like this. This dimension, let us make it like this. This is cross section is 1 micrometer, right? Right? Now this length, 1 micron. So this is the cross section. Now I have a pore. It is not even this much, 1,000th of this, and they claim that this can study 1. It can give the flux ion. See, what they are doing is patch clamp technique. What it does is, this is studied. They go and place it with respect to, now this is the cell membrane. They'll come and place the electrode like this. So the electrode has got a mouth. So that mouth, there is a channel. This is a channel. The mouth of that will go and cover like this. So if this is a nanometer, I should have at least 5 nanometer, I can agree. 10 nanometer, I can agree. It will isolate one whole bacteria is only 1 micron dimension, and that my bacteria has thousands of ion channels. How can it isolate one? Did you understand basic concept of dimension? No, you didn't understand now? Because this is a Nobel citation. I don't understand how they can cite so openly. It will go off. It will go off. This citation will go off. They will remove the text. You go and see Neher and Sackman. You draw. Better. My 3-dimensional depiction is. Yes. You remove that. Yeah. That's a bacterium, or that is a cell. Imagine that's a cell. There are multiple pores here, thousands of pores, right?
So the thing that sir was saying, simple. So you have this probe. You put the probe. If you are using this probe to study single channel, what is the width of the single channel? 1 nanometer. Now if you put that probe here, how many
channels are you covering? Thousands of channels. How can you say you are studying one? Can you even understand what is happening? Through the channel, things might be coming inside, might be going outside. And at the end of the day, of the experiment, you will have a net, if you say something that has come out. But then you have also lost something which has gone inside.
So they're studying the current. What they do is they study the current, and when this is used to study the current, the earlier technique was used to study the potential difference, right? Now I'm saying, what the hell? The neuron is conducting from here to here. What do you get by a potential difference measurement? You should see something going from here to there, and this is to enforce their own study. I'm a simple guy. You tell me all cooked up stories, 100 people, 1,000 people agree, obey, and all that. No, it has to go through with my basic idea of dimension first. If you tell me that I have put 1,000 elephants in a this kind of space, கடலை சாப்பிடும்போது அதுக்குள்ள என்ன சாப்பிடுறப்பா, நான் யானையை சாப்பிடுறேன். When you are eating peanuts, you cannot tell me that you are eating elephants like that. So dimensions don't agree. The claims, why? Because these kind of things can be only had by extraordinarily high amount of money. The extraordinary high amount of money can be had with only very powerful connection. That high powerful connection is a catch-22 vicious loop. You have to please the people who will sanction the grant. Then those will sanction only if they are in the ATP synthesis. They showed with single molecular experiment that actually ATP goes round and round. How did they show? ATPase, that protein goes round and round. How did they show? They took the protein, they attached an actin filament, and they attached a fluorescent probe, and they changed the field and made it go round. I take a knife, put it into a pumpkin, I put a a sail on the handle of that knife, then I blow a wind turbine or something, and first I shake that pumpkin really loose so that the knife is sitting all loose. Then if I drive it, it will perhaps go round and round. So, what is the point? Yes.
Can we use now nanopore technology type of thing to measure this?
Yeah, sir. For me, this, if you want to study this, my point is the relevance of this system comes while we are observing. Now we have a nerve, right? Why all these systems are important? These systems are important to understand how a nerve conducts an information from one point to another point. That is, now you have a nerve, so this is the nerve that collects information from here and goes through a long procedure. So your spine ends here. From the foot all the way until here, you have a single nerve. You must understand. So this conduction of that information is from here to here, in real space, in real time. How they say it is, this is going sodium inside, potassium outside, sodium inside, potassium. What kind of nonsense is that? Yeah, it doesn't make any sense. I will tell you, I will cut my own head off if this is true. It cannot be true. It is not true. People have cried out against this sodium going in and all that. They saw, if you just change the sodium ion into trialkyl some ion, organic ion, you still get the effect. What the hell? Where is your channel specificity and all that? Nonsense. Anything, you remember this? They forget all this. They will always see this. You will keep reinforcing your false beliefs. It's sad. No, you just use your common sense. If something has to go from here to here, is it a ripple based effect? That's a ripple, some kind of ripple. So what it says is, I'll show you that image. You know, this is the sad state, how they gave them Nobel. Voila. I should have, where is the better, can we just Google, the Google has got a very beautiful, you can go and see. What happens is, this is the, yeah. So if it is real three-dimensional space, they have shown the how temporally the sodium and potassium channels open. When you put what they did experimentally is, they put an electrode here and they put a reference electrode here outside, and so you are now measuring voltage here, right, across 2 points. That's what they have shown. When an information passes from here to there, they observe this, and for explaining that, they are saying in real space and time, it is going like this. Suppose now from here to there the information is going. Now some sodium potassium now here sodium channels are open, and then potassium channels are closed, then potassium channels are open, so sodium channels are open, then potassium channels are closed. How? Why? How and why? They say these are controlled by the potential, these openings. And then they say the protein controls the potential. This protein controls what is pumped out, and that potential also controls. How is it possible? It is a mirage. Just like that proton pumping was a mirage there, this is a mirage. Now, if I pump something out and I create the potential, and the potential is controlling me, I mean, where is the logic of why should something open and close at one point and close at the other point? See, I told you, sodium opens here, there sodium is closed, potassium is open there, like that. That is how it is supposed to go, it seems. And then sodium is going from here to out to in. It's a very simple logic. Something goes from here to there. That's it. It cannot be anything else. So now what are the alternate theory? It's a sound wave that travels. It's so many there are alternative theories. There are no, actually that's a better theory than this theory. Phonon, there are so many other explanations. It's a far better explanation than the Nobel recognized theory. So I'm sure, because
now how do you explain this one?
Now my theory is, he asked me, where does the electron go from here to there? I say that it can go either on the outside, or more probably it is through the microtubule. Very defined structures are there. They are electron. See, because you see these proteins, they were not redox proteins, sodium potassium ATPase, right? But in sodium potassium ATPase, just like lactate dehydrogenase, we have shown that they are sensitive to a wide variety of, where did it go? Yeah, last, yes. See, all these kinds of multiple redox active molecules, I've just shown 2 examples. Now, you imagine how this nerve conduction, you have chloroform, or say, when you go to anesthesia, they give you haloforms, basically organic, gases. Now, very simple. My theory is these radicals, they hit this organic halogen containing. You see, in cytochrome P450, iodobenzobromone was more effective than bromobenzobromone, which was more effective than chlorinated benzobromone. So why? Because that's all radical reactions. You have seen, what was the first example of radical reactions in your chemistry classes? CH3Cl. Remember those reactions? Yeah, that's it. So basically, it's a radical chemistry that when you are exposed to this, it hits it, the Ross dynamics gets altered, and then when the gas vapor pressure comes down, the system is restored. So it's an electronic circuitry. It is not a ion pumping machinery, channelizing machinery. It's a very clearly neurons are electron active things. It's a very simple system. That can explain why when the myelin sheath, when some sheath is here, the relay is much better. It is faster. If it is an inside-outside activity, the covering will only mess with the propagation. Yes or no? That's it. So, fundamentally speaking, so now you have seen that cellular activity, what is the most fundamental activity of a cell? Powering, then coherence. One thing happens here, another part of the cell must know that something has happened there. It should act as one whole unit. So this electrical connectivity can enable that. Then powering, coherence, homeostasis. Water, oxygen, solute maintenance is a equilibrium. So when something is formed, water goes up, it creates a pressure, it can lead to some opening. Simple like that. So it can maintain some concentration like that, and then water gets into equilibrium. So powering, and water can, once you have a higher amount of some polar things formed, it will take it out. Osmosis and all that, there are ways of doing it. Then powering, coherence, homeostasis, PChems, electromechanical activity. Tomorrow you will see how electromechanical activity, that is how this oxygen oxygen superoxide, ta-tan. अरे यार क्या कर रहे हो यार? They have done it so many times already. That is why all this is. And this guy tells me that, sir, when they want to disrupt the class, they kick the water. Yeah. So what was I saying? Yeah, electromechanical. How this oxygen superoxide, it will give water formation. That water formation will change immediate space phase characteristics. So water is not a free solution as people said. That is what Professor Yahya was saying. That is what Professor Lawrence Jakin was saying. People say that water is a solution. All noble their equations. Hodgkin-Huxley's is dilute solution equation. People have struggled against it. Visually you can know that it is not dilute solution. It is a colloid. It is a colloid. You could break open an egg. What is it? Is it a solution? No, it is a colloid. So basically, common sense has been violated because in 1930, one Nobel laureate arbitrated a dispute between physicists and biologists by finding the distribution coefficient of urea in cellular cytoplasm and water. He found it to be 1. So he said, okay, it is like normal water. Water inside, water outside. There is no difference. So that's why you see Hodgkin-Huxley's equation, for example, is starting with solution chemistry. This is solution chemistry. And that also is a miracle. They explain all this because temporarily they change the permeation constants. Is there any evidence for that? No. What that mathematics does, sir, you will understand what they are doing. See, what they do is they are getting this potential variation in time. How they are explaining is that the membrane channels have different selectivity at different times. That's it. And you put constants and all that, they will explain. That's it. Is it ascertainable? No. Is the voltage? See, their theory is like this. The membrane protein generates the potential controls the protein. I cannot imagine how this explains anything at all. It doesn't explain anything. I'm sorry that I'm appearing so neurotic and all that, But the point is that how can so many thousands and thousands of people across so many continents be taken for a ride like this? Yes. Yes, sure. My talk has ended. I have no clue. I will not try to go there. I'm colorblind partially myself, so I have no clue there. Colorblindness. It's a good question actually, because we have rods and cones, and there are 3 types of cones, and we are supposed to get a mixture of that and effect. I try to read up and try to go into it. I was not really much into it, but that's a very good question. Yes, I see. The brain is a wonderful thing. I don't know whether it is the brain, but mostly it looks like the brain, because some part of the brain is supposed to be involved in that, and they even if that part goes, other part also recovers that ability to sense that. So yes. Now here the chemistry and the timing, all that very beautiful structures, all agree. Sir, first, 1 second. My boss.
One you said it takes one cycle takes how many hours? How you are studying that using your theory?
Sir, their theory is the retinal pigment epithelium. Yes. Here, this thing, sir. Now you see here, one photon comes, binds, and this cascade pops out this main thing. Now this has to go through. It has to come out of this cell. It has to go permeate into the retinal pigment epithelium and come back. So this guy is rendered ineffective. So basically things like that. Sir, that is the minimal, sir. I mean, what I'm saying is it's got multitudes of problems, and they themselves say that the kinetics don't agree with whatever. The point is the chemistry doesn't agree, the kinetics doesn't agree, nothing. So here they don't say, in the bacterial system they say rhodopsin is a proton pump. Here they say it is somehow popping out, all that. Murburn theory, very simple. The molecules inside, outside act the same way. They don't feel, okay, now I have a membrane around me. Now I'll live. I'll have my own wishes. I'll have my own fancies. I will not do that. This idea, the oxygen superoxide equilibrium, is the most facile one. Yes. Now, sir's question address, please. Yes.
From photosynthesis I am coming.
Yes.
Because when we are studied about the fluorescence, it will absorb all the different colors. That is what thrombosis we just color connecting them. It is reflecting.
No. Chlorophyll cannot absorb all the colors. No, sorry. There is something called.
The green color.
Yes. There are certain things. Every molecule, if it is colored, it has its own absorption spectrum. My dear, you can talk amongst yourself later. There's a teacher who has asked a very relevant question. You have no clue what she has asked, right? Yeah.
It is about the reflection from the fluorescence, because it is absorbing blue and red color at the back, and it is reflecting.
Reflecting. Use another word.
So that is why we are observing it in the green color. So it's the same way it is having the carotenoid-like pigments.
Yes.
So is there any? That is why the reflection is observed.
No. Reflection. Don't use that word. Let's use the right word, not reflection. Okay. So you say the color. You are addressing his question. Yes. Part. Okay. So let me tell you that the rods and cones are supposed to be black and white, gray differentiators, and there are 3 types of cone cells. And for me, by what I saw, the pigments, all of them are melanopsin, rhodopsin, simple pigments. For me, I don't know. For me, the color, I try to read up, I try to analyze, it just avoided me. So whether in photosynthesis, you see, photosynthesis also, there are carotenoids. Same molecule there. Same molecule. Why? And my theory says, in the murburn theory, it says, in the photosynthesis, there is nothing called exciton, transfer, relay, and all that. That also gives ROS. This also gives ROS. You take it, you put it in solution, hit it with light, it will give ROS if there is oxygen. It is a simple chemistry. Why are we trying to complicate things when there is a simple explanation? Why does the protein have to change its shape? Why does it have to go from here to there, hold some proteins which it doesn't know, then come back? Why? It is explained with simple, bimolecular reactions. These are sequential. Now, I didn't tell you this. This one thing. Yeah, this step. You know what is the probability of achieving this? Even if you have 1/2 for binding, non-binding, and the sequential, 1 by simplest is like that. Unbelievable, yeah. Unbelievable. I mean, please record this. See, please. Unbelievable. You guys, if you get to see it, hopeless. I'm telling those people, my opponents. Yes, sir.
One research from Kerala University, carbon suit.
Sir.
Carbon suit. No, what is that?
Carbon suit, that black thingy.
Black thing coming out of now.
1D.
Yes. And it is put on leaf. Increases the photosynthesis. How they explain that one?
No, wait. No, definitely. The chemistry would be there. See, I don't know. I have to think. But then, my theory is beautifully explaining this laser therapy, photo laser therapy. What is that? Photodynamic therapy. Yes, that was the word. Photodynamic therapy. So photodynamic therapy, when you put that light, it gives ATP inside. Why? ROS. The light, there are so many things that absorb there. It can also give ROS. You get flavins, so many color pigments. It explains somehow some aphids can do photosynthesis. Aphids have collected some genes and all that. Aphids live on plants, and they have some what is that called? What kind of horizontal gene transfer? Is that horizontal gene transfer? Vertical, horizontal, whatever gene transfer. I don't study horizontal gene transfer. So horizontal gene transfer, it does. It's a plant photosynthesis. How? Something it can produce with ROS. It doesn't need to go through the Z scheme. Photosystem 1, photosystem 2, nothing. You don't need. Water can be broken by simple molecules right in aqueous chemistry. Why so complicated? Why is see that manganese complex that is there? It is just a peroxidase-like thing in cytochrome oxidase. That is exactly that. That's exactly what it does is it pulls, because of the multivalent manganese, it does that. That's all. It is not so complex. It is not at all complex. The same, if you just accept that diffusible reactive species are obligatorily involved in cellular, chemical, electrical, mechanical activity, everything fits the picture. Only thing that stops you from accepting that is decades of indoctrinated belief that ROS are bad. When you exercise, you get muscles, your activity, all that goes up. ROS goes up. So, ROS is bad. Why? Everything. So, nitric oxide, it's a molecular messenger. What does Viagra do? It increases the blood circulation. How? Nitric oxide is a ROS. It is a radical. Nitric oxide is a radical produced by arginine metabolism. It's produced by an enzyme, and that's how it is communicates. That has gotten a Nobel, because the guru said it. This is explaining all fundamental processes. ROS cannot be avoided. You see all those leaves parts which have high amount of ROS, or all those leaves which do high amount of photosynthesis, they die quickly. Why? You burn more, you die soon. You burn more, you die soon. That's it.
That photodynamic
Therapy.
It is also conjugated with some medical, some molecular, drug molecules. So that also explains some release of electrons.
Yeah. Maybe, I don't know. So the point is, so what is the time now? Can somebody tell me the time? 6:40. Okay. So we'll start at 7 sharp. Okay. So now, what have we seen till now? Can somebody take a mic and pass the mic on amongst you? Until now, we have spent 2 days. You have seen an overview, examples of some systems. We have seen today complex systems, right? How unknown molecules are metabolized, how energy is produced, how sunlight is tapped, how we can perceive light. Yes. What was the single element that can explain all this is oxygen superoxide equilibrium. Oxygen superoxide and water equilibrium. Right? Oxygen superoxide, it doesn't stop there, peroxide, then hydroxyl, hydroxide, and then water. This equilibrium. And that is why we need oxygen. It is not because oxygen just makes water, it is these things that are involved. Without that, it doesn't. So, what are the interesting things? Just pass the mic on and just speak for yourself. Just say something. Each one of you. Ayyo, என் கையில மைக் வந்துச்சு, எல்லாரும் என்ன பார்க்க போறாங்களே. Sorry, don't know torture. I will not torture. Ayyo, I'll go and I'll bury my head under some. Yes. Doubts always take precedence. What's your name, my boy?
Hariharasudan.
Hariharasudan. Yes.
You thought about the vision of eye. The vision has another 2 conditions, which is myopia and hypermetropia.
Yes.
In those cases, the light fall on retina. Those are the things. For that thing, the reaction is same for every conditions or differ from each other.
See, my dear, the hypermetropia and myopia, that is the image falls out of focus, right? So imagine it is falling out of focus. What the current theory says is that the molecule, the photon should go from one after entering, it should go 10 different layers, it should reach there. That's impossible. So what I have said is that multiple layers. You know what happened? Last year, there was a paper published in some advanced cell high impact factor journal, and they said multiple layer processing of visual stimulus. I said, dude, Vivian and I published the paper in 2020. You have not cited our paper. We published in 2022 in JCP. We have not cited our paper. We wrote to the editor. They didn't respond. So the point that I am saying is, it falls here, there. That is what happens. That is primarily because of a lens effect. All right. And so thereafter the processing is hazy if it doesn't reach. That is acceptable. Here, you should imagine that for reaching, if this is the eye screen, what you are asking is the lens, it passes through, it falls here, it might fall there. So it might be out of focus, so we will not get it. Now, if you have a perfectly working system, you have a single photon, it comes here, it has to go through 10 different layers, diffract, scatter, interference, so many phenomena. So in one time it will come here, in one time it will go there. How can an evolutionary procedure ever come around that can see with that kind of a mechanism? That's what I have highlighted. That's what Vivian was talking about. That's what he set out first talking about. Because that is something so silly. Anybody should ask that question first. He said rods and cones are the primary phototrans receptors. How can it be? See, 400 nanometer, one photon, 400 microns is the thickness. This is why I say elephant, car. You should always understand what is the dimension of a atom, what is the dimension of the I mean, you know, all that. Basically, the point is, if you say I was eating 200 elephants from this Kerala, like this, you know, like this, nobody will buy it. But these kind of things are being sold here. Of 1,000 times its wavelength. Cannot. So that is the primary thing. And then multiple questions that were put up in the slide, right? If you remember, why do you have blackouts when you have oxygen shortage? Things like that. Basic, a theory, what is a theory? An explanation is something that makes sense of starting assumptions, ending observations, whatever we have, nothing. And these are the most fundamental processes that life depends on, that your body, you know, it cannot be. So yeah, pass the mic. Somebody should be able to give some feedbacks now, because otherwise this whole me talking to you and Robert De Niro is saying you talking to me, doesn't make sense. You know, and half the people didn't get it. So yeah, where's the mic? Yeah, pass it on. Give it to that guy. ஒண்ணு மேடமா சந்தோஷமா இருக்கியா? ஏன் சார் என் வாழ்க்கையை கெடுக்குறீங்க? You want to say something? You want to say something? Nobody. Who wants to say something? Kiddos, these are the times where you start expressing, communicating what message I sent you. See, yesterday I did not sleep. You saw something? Why? I could have easily slept. I had n number of things I could have done. Here. Interact here. Sir. Learn from it. We got that. One second, sir. I'll give you the mic. 1 second. That thingy we were waiting. Sir was there sitting at 9 o'clock. We got that. We wanted to put here. How many people have seen that? Why have you not seen? Okay, so the point is, we all worked to get it quickly, and sir was sitting there. He was seeing this, he was seeing that. Sir, please move. Sir is not moving. He is seeing. What did Vivian say? That childlike curiosity, that interest, that zeal to communicate as a social entity. That's what our scientific temper is based on. Sir, please.
Here now, or if oxygen is involved.
Yes, sir.
Where does it get oxygen?
Sir, that is why, very good question. Now, the eye, retina, it is innervated with blood vessels both sides. Why should something be copious blood vessels? Why? Because oxygen is the thingy. That eye, retina, ipratum, apratum, high amount of blood vessels. So you see the structure. Oh, that's a very good question. That's important. You see, now, oh, we did not cover one thing at all. Oh, this slide, very important, very, very important. Now, this is the structure of the eye. Multiple layers, and this is the layer that they say is getting it. So, there is blood vessels here, there is blood vessels here. Now, why should it go all the way through? If it was a simple setup that could have easily evolved, I would expect the rods and cones to be not like this. The orientation should be towards this. Rods and cones are here. The discs are here. The stacking is here. It could have just faced, and the you could have had the very simple setup like this. Blood vessels would not have interfered. You could have connected, and the nerves could have just connected from here and taken it. Now you see, it comes here, all that is coming, and the nerves, how does it, you see the structure of the eye. It's very interesting. You see this? Here, it has to pierce this is a blind spot. It is collecting the nerves all, then coming like this. So the electrical signal that goes, it originates from bottom, goes up, and then comes back like that. Why? We should have had a simpler system wherein it goes like this. Why should it go such complicated, nonsensical thing which doesn't make any sense? That's the reason. So, yeah. So when you sit there, something should, come into your, something you should express. You feel like communicating. Mesla? Anything? Her brother called me, says, "Sir, please take her." I said, "No, registrations are closed." Mesla came here. She is doing PG in Government Victoria College, right? Palakkad. Yes. And she has been sitting. She did not ask me any questions. She did not like me, it looks like. Or people, anybody, you don't feel like asking any doubt? Illa, yanakkellam purinju pochu. Yenna Yes.
Any vein cutted in eye means that time the light will be enhanced.
Any vein?
Any
cut,
blood vessels
cutted
means
cutted, like cut. So
that time they analyze the light. It is
no, take the question somewhere. Take the question somewhere. Any vein cut in the eye, light will be enhanced in a purila. Did you understand?
I think he was earlier asking about blind people,
Mm-hmm.
and the cutting. If there's any damage to the blood supply to the eye, how do they go blind? That clear.
How do they go blind?
How it is going to affect the vision?
Yeah, basically that's what the first question written there is. When oxygen is low, you black out. You cannot see. You cannot see. Oxygen goes down, you black out. So that's one direct signal that.
That happens in the case of anemia.
Yes, it's very oxygen. Yeah, you cannot go. You black out. You don't see anything.
The vision becomes blurred.
Yes, and you have too much light. You cannot see. That's because too much ROS. There's an optimal amount.
And the cells would die if oxygen is not established.
Yes.
Soon.
Soon.
So permanent loss of.
Permanent loss. See, oxygen is bad in high amounts. Oxygen is bad in low amounts. Why? This whole thing is an equilibrium. You need to understand interactive equilibriums. That's where our science is going. That's where murburn will take. The next, the 20th century was of that gene and computers. 21st century and the rest of the millennium, you will be trying to understand how these things can be controlled or understood and controlled. Right? That's where we are going. Hopefully, sir, boys got 83 accuracy and things like that. They're very, very happy. Yeah. They used some transformer and they got better results. Now they are doing something else. Yeah. Tomorrow, if I can get the NIF director to press a button and show something like that, that's what we are. Some amount of political stunt also we have to do, if possible. Yes, 7 o'clock, yeah. Yes.
5 minutes.
5 more minutes.
It is about, I already discussed with sir.
Yes.
This ROS.
Yes.
At very minute concentration, it plays an important role in the activity of this one. And if their concentration increases, the enzymes are acting over there, that is the peroxidase or catalase or superoxide dismutase, whatever the scavenging enzymes, they are involving in their conversion. Even at a very higher concentration, it acts as it is suppressing the activity. So what is the concentration in that particular? I am segregating into 3 categories. That is, one is at the toxic level, and one is the area where the scavenging enzymes could act on that one. Below is the area where.
1 second. பேசுறது கொஞ்சம் கம்மி பண்ணு பாரு tone. Yes. No. I'm telling him. இங்க கேக்குதுப்பா. Yes. I'm getting distracted with that. Yeah.
So what is the concentration of these 3? The third one may be at molar concentration or less. So
yeah,
what is
good.
The concentrations of these 3?
Wonderful question. If you were attentive, I had put in chloroperoxidase, I had shown you the order of dependence. That is, I had shown several experimental plots wherein I had shown if there is an active site mechanism, how the outcomes, suppose this is activity, you remember the active site reactions were like this, whereas some radical mediated, ROS mediated reactions were practically like this in micromolar ranges. So now you want a system to work in a rather digital manner. That is, from this range to that range, you wanted it to give the same output. What do you want? This cannot be given by the analog system. This is a digital one. That's why when Phil Penketh, he's an American researcher who was working in, which university was he? Very high, Cornell, I think Cornell. He was in Cornell. For 24 years, he tried to publish one idea. He got 24 years. Finally, he published that paper, giving $3,000. I felt so bad. He's a very smart, very sharp guy. Yeah. So the point that I wanted to say is, here, at these micromolar ranges, you will have very low activity with active site, because the species cell have to diffuse into the active site, which is kinetically challenging. So now, if it is reaction occurring there, see the concentration. So at this is the substrate concentration, right? Substrate. Now, if I were to the DRS concentration. So when the DRS concentration varies, this is the plot. So basically, this is practically 0th order with respect to the substrate variation. You should understand what an order is, what molecularity is, all that. These are basic kinetics aspects. You should also understand some amount of thermodynamics. I'm not asking you to understand a lot. All right. Don't get scared and run away from that. You cannot understand biological processes if you get scared and run away. It's very simple. All that you need to do is, if you understand once, you will never forget. It's like learning cycling or swimming. You will get that balance. Right? If you try to rote learn, it will not work for you. So this is the question that you asked is very good question. Very good question. So why are some reactions like this needed? Because the cell, I need to keep working in a particular way, in a standard way, even if some molecules are varying, right? It will give one, because this is the primary thing that is operating. So if it is lower, it will give lower activity, but for the substrate it will not. But the active site binding based will give usually in that range, hyperbolic fit, usually Michaelis Menten fit. Did you appreciate what I'm saying? Your question was, in which range, millimolar range, is it bad? Micromolar range, is it good? It depends upon different environment, what the ROS is, all that. We are just at the tip of the iceberg now. This is going to unleash a huge change. Only thing we have to undo is this decades worth of aesthetic, stigmatic perception. Don't get psyched by aesthetic, stigmatic words like that. Sometimes it comes in my mind that when Govindji said, bombastic, that words, this words, it comes. I cannot help it. Yeah.
Okay, so as a biochemist, when we talk about enzymes, we focus a lot on kinetics and how to manipulate kinetics. So how does murburn concept affect kinetics and the study of kinetics?
Oh yeah, that was first in Laurent Jaquin's talk. That's the first thing he said. Atypical kinetics. So if you saw with some attention the earlier talk, wherein no, not the earlier one, the lunch one. No, yeah, the dose response one. Yeah, the first one, drug metabolism talk. There, see, what is a normal enzyme kinetics? That is, enzyme binds with substrate, forms ES complex, and then E plus P. Now this they subjected to mathematical simple some assumptions, and then you put rate constants here, rate constants there, subjected to equilibrium, and then solve. Then what you get is, if you put rate in the substrate concentration, wait, I'll come to it, 1 second. Now as you increase the substrate, the rate will go high. More and more you increase the substrate, what happens? Why? Because substrate is binding at one unique site. After there is finite small amount of substrate enzyme, you increase the substrate more, what happens? It will start saturating the enzyme binding site. So as it starts saturating, you get the asymptote here. All right, this now Km, as per this, the substrate concentration at its half, there are certain features to study. This has given some ways for you to tangibly understand and talk about your work with others. So this gives a very good tangible way of dealing with the subject. Now comes murburn. Now murburn, guys, you might get ting tong, something like that. What will they do? They'll plot something. They'll happily see this part and that part. They'll put R square values here and there, and they'll be happy. We have been closing our eyes to data. So I don't know why it goes here and there. I can tell you there are mixed interactions and different kinds of. I put that mechanistic things. What exactly? We are also trying to. Sai was saying that we should nail this. I tried doing this. I tried to mathematically do it. Basically for that, if of course there are lots of software which can do it, but for me, I don't know this. For example, one thing that we have is now multiple substrate. Earlier in the Michaelis Menten this thing, so something like this. Now, if it goes up and then comes down, what they said is, in the active site, suppose this is the enzyme, this is the substrate coming and binding, then there is another binding substrate. So what happens is, this can react, but it cannot go out because this blocks it, right? So that is the reason why at a higher substrate, it gives that. Now, the same enzyme or different enzyme with multiple substrates also show the same property. So how likely is it that it's a binding based field? It's highly unlikely. So if you invoke to understand and appreciate that kinetics and how, what we are doing is, we are just trailing the molecule of interest, that product formed, and that is going to do this. If you see the whole reaction system as such, you will see multiple other molecules are being formed. That I have shown in multiple systems. So, അങ്ങോട്ട് പറഞ്ഞു കൊടുത്താ അങ്ങോട്ട് ഒന്ന് പറഞ്ഞു കൊടുക്കാ കുട്ടിക്ക്. നിങ്ങളൊക്കെ ഇരുന്ന് അവിടുന്ന് ഇങ്ങനെ വണ്ടി കൂലി വന്നിട്ട് ഞങ്ങൾ ഇവിടെ തീറ്റയും പാർപ്പിടവും ഒക്കെ തന്നിട്ട് നിങ്ങൾ പരസ്പരം ഇരുന്നിട്ട് ഇങ്ങനെ കൊല്ലാൻ വേണ്ടിയാണ് കഷ്ടം. I have to do this. You are not breastfeeding babies. You are listening to science, the latest aspect, and when you don't know the value of it, it's sad. It's sad. And I will never ever let it go like that. I will never ever let it go. All teachers should not allow. They should be bloody constipated like me, I say. You should not allow that. We should not allow that. Yes.
One follow-up question.
Yes.
So, what would be a good way to, to do kinetics of Amur science? Would it be good to accept all possible products?
Yes. One way to do it is to trace it with LCMS, for example, or GCMS or something like. If you need to be seeing you've got a mixture. A very, very nice question. Yeah, I like this. Nice question. So now, then but the point is now, how does an experimental system work? The handling of it. Usually what you do is, when you have an experimental reaction, you either follow the substrate disappearance or the appearance of a product. Or if you have really a sophisticated lab, what you do is you try to follow both. Or till now, people, multi-product reactions, they are not really interested because they like predictable, tangible, deterministic outcomes. So ideally, when you try to follow something, some molecules will be highly polar, some molecules will not be that. So some ions would get reduced, oxidized. You'll have to have a wide variety of things to really deal with all the products that are, all the things that is happening in murburn. Because fundamentally, if you want to say, I burn a paper and I want to follow exactly what happens, and the paper has got one blue line here, one black line there, and all that. So it is burning like this, like that. This time, next time you set it, it will burn like that, like this. And the temporal area, it will all go a little bit here and there. So I'm sure we will, with AI, it will get nailed pretty easily, because in spite of all these complicated things, we are getting reproducible, very beautiful data, outcomes. And it is sure that we have survived for whatever matter. Our kids are also going through the same growth curves and all that. We get very reproducible. So, and you should see the data when we get kinetics, when we do. With NADH disappearance, it's purely a one electron disappearance chat. Even into micromolar level, the chlorination reaction and the NADPH reaction, it comes like a scale which you draw. Absolute. That is basically what I'm showing is, it's zeroth order. Basically, beautifully goes. So predictable. Enzyme, you cannot get it like that. This is activity, or here it is substrate disappearance or time, for example. This is kinetic measurement. So, point being, when you are trying to see an experimental system, most of you will end up as experimental scientists. My life, my works, all are based on one simple observation. Or, some peroxide, I told you, it doesn't deactivate. And then I thought, hey, I can show you that. In fact, before, the day before, I was working away, we ordered some chemicals, and because of this presentation thingy, that thing, we got the peroxide, we got the thing. I did not get the enzyme. I went to the shop and bought a radish, smashed it up. Muthu is here. Yes, we did that together. Yeah. Yes, the peroxidase activity. The enzyme did not come. So what? We got a natural source. Yeah, and we did it, and we usually 10 millimolar it inhibits, and 10 micromolar we usually get a spike, but I did not get it. So this was a different enzyme, this was a different peroxide and all that. So what I did is I did a serial dilution. So here was the outcome of that in the simplest way, the murburn, to establish murburn. That is, I diluted azide N3 minus, and see, I hear that when I hear that chatter among themselves, you can go if you don't want to be here. Go to your hostels. Okay, I cannot tolerate people talking loud. So 10 to the power, say this is 100 millimolar, for example. So 10 millimolar, 1 millimolar, 100 micromolar, like that. Now from this I take and add into the reaction mixture, and I put test tubes like this, and I see this is control, no azide, right? So this is, say, 10 millimolar azide, and then 100 microliter I add into 1 ml, so 1 by 10 dilution. So this will be like that. So like that, 1 millimolar, so 100 micromolar, like that. Now when I did that, here in the control you will get high activity X. Okay, now in the azide containing, high azide containing thingy, you will get, say, 0 activity, no color. And then when you go down and down and down, it will try to get control activity. And then one point will come wherein it will show higher activity than the control. I can show you that. So this is something that definitely cannot be explained by Michaelis-Menten. It cannot be explained. So that we did it. We have got the chemicals. If you want to see it for yourself, I don't know how people, because that takes time. Serial dilution, making it all takes time. So if you are interested in experimental science, and I can show you the demonstration of this. We did it. Finally, we had to make a serial dilution, and because every single system, it will be different. But then this will be reproducible. In one system, it will be 10 micromolar. In one, it will be 1 micromolar. It's a rough age. So that is what, certain molecules in certain systems, when that gets stabilized in that kind of environment, you get a drug. Now you will have murburn drugs. Actually, I think metformin is a murburn drug. The antidiabetic drug. Because it has got the biguanide. Biguanide is highly electrophilic. Yeah. Once again, Judy Hurst, John Walker's group, the Nobel laureates, they published a paper in Nature. What they did is, this biguanide, they derivatized with a long, bigger phenyl moiety and all that, and they showed that it goes and binds to complex 1. So basically you have what do you call, in Malayalam, aniyati ganicha ammayi kattike. You understand that? No? What it is like, your molecule is a small polar. I have that, and then I attach a huge thing to that, which is hydrophobic, and that I put a ton load of it in the enzyme. I crystallize it because I have a lot of money and I have a mandate, and I show it. If you want to show anything, I can show. You want to show, I have a theory, I want to prove it, I can show. But I will need it unrealistic things. Now with murburn, what we can do, you see one mitochondria kept here. There was a paper published. One mitochondrion kept here with another it introduces effect in another mitochondrion here. How? ROS produces enough energy. ROS reactions produces enough energy. Energy can go through, and that can stimulate activity here. If you just believe the basic glycolytic pathways and all that, which have delta G as 5, 10, 15 here and there, and that kind of radiation will not be produced. So all these inexplicable aspects, you can easily nail it. Wonderful. So I think we'll call it a day for you today, because the people there, they might have respect but not interest. Yes. Yeah. Thank you so much. Tomorrow is the last day. Tomorrow we will have what? Yeah. That was pretty. अरे हाथ दे रहे हैं यार, हाथ क्या दे रहे हैं? हाँ अच्छा, ऐसे नहीं, वो हाथ दिखा रहे हैं, टच करो यार. No, that. So the point is, touch is very important. When somebody reaches out, touch it. Don't be saying that. So tomorrow in the afternoon or in the forenoon itself. No, forenoon, I don't know. Around noon. Afternoon, I don't know how it is going to come, because the last discussion we want, that is what is focusing on our school. We'll try to get how AI more. But now you got some idea. Now we'll have a quiz on murburn concept generally. All right. Now we'll see. I'll try to give you a merit certificate. It will be just for bragging rights. Not that you can do anything with that for now. When you get older, perhaps you can show. All right. Okay. If you really value the process of doing science, and
but now if you show it to any institute, you will not get the entry.
Yeah, that may be true. But no. Who is this? What is this? That's what it is. All right. There are lots of people. There are lots of papers, and no. There are also people who like the work. They'll say it in private. For example, when I went to Steve Flyger's lab, now there's a professor there. She came to me and said, "Please don't say this outside, but it explains everything. Don't say this. I want a tenure here." Tomorrow we'll have multiple areas: evolution, immunology, every other thing. Parallel medicine, this, that. It is going to be halwa tomorrow.
Unconventional, non-conventional.
Yes, non-conventional medicine. So many things. So can you imagine how murburn can now, basically address so many things that you can see in immunology, in so many things. Okay. Now I think I said 7. What is the time now? 7:10. I think we should call it 7:17. Yes, I think you guys go. And how many of you are going to stay back tomorrow? Okay. Yeah, you guys are. There are a significant amount of people. So no dinner tomorrow. No breakfast day after tomorrow. This is like basically for you. Huh? Dinner kodukano. Sir has said dinner, give dinner. So I can tell them, they said 12 hours notice, I can give dinner. So, okay. So, say thanks to sir. Yes. So, yeah, thank you so much. You have a good night activity or sleep, whatever it is. Bye-bye. Thank you, sir, for coming. Thank you. Thank you. It really means a lot.
Radioactive materials, they don't follow kinetic.
They don't follow anything, Surjit. The point is to expose them to some amount of original thinking.
I want to know, if I put superoxide in water, if I put superoxide in water, expose it to some light, whether it can produce hydrogen and oxygen.
Hydrogen and oxygen, light, superoxide. I don't know. You have to see. The point is that you need to have some immobilization or localization system, because the superoxide must be localized and some kind of stabilized. இல்ல நீங்க 2 பேர் இருந்தீங்க. No, it means a lot to me. It was okay today, no?
Ask them, sir, not us.
No, I want to be safe. Imagine you make so much preparation and they say, oh, sir, it was, I know my voice. They'll not.
Of course, you should not try to.
Yeah, yeah.
They want to know at what time tomorrow it ends.